Thank you for the invitation to talk from the organizers. My name is Trevor GrandPre. I'm a assistant professor at Washington University in St. Louis. And today I'm going to talk to you about trait space patterning. Uh, and that might seem like a lot of jargon at the moment. So I just wanted to go through this a little bit more slowly, just introduce this is phase separation or some kind of patterning on a two-dimensional phenotype base. So this doesn't make any sense, but this is sort of like where I'm going to take you. But first, I want to just introduce eco-evolutionary systems and what that means. So generally, eco-evolutionary systems are ones where ecology and evolution are on similar timescales, and there's feedback between them. And this is actually very common in many biological systems. One is, for instance, is predator-prey models. So, for instance, a guppy fish have many predators. So because of that, they actually grow to smaller sizes, even without the predators. So this is the feedback. Another sort of big model could be our gut microbiome. So whatever we eat affects our gut microbiome, but that also changes, the feedback on how that on us and our health. A third of many is bacteria and phages, where they've been co-evolving for billions of years, and it's been an evolutionary arms race to, evade each other. And so this is primarily the system I think that's most relevant to what I'll talk about. So maybe this is like the context to think about the results, which I'll continue to introduce. Okay, so this is eco-evolutionary systems. And so let me just keep kind of describing this idea of mutations now. But yeah, okay. So the idea is that traditionally mutations have not really considered, or models of mutations have not really considered how mutations occur. So, there's many different ways you can get mutations from the environment, from replication, from many, in many different ways. But traditional models don't really consider these types of modes, what I call modes of mutation. Instead, traditional models treat it more of like all of them as the same up to some time rescaling, and really it's just like a constant or some probability to change that an allele. So for instance, we could just as an example, you can consider for a gene, there's two alleles, A and B, and with some frequency P, you have A, and with some frequency 1 minus P, you have B, and there's a fixed population size. Then, one model that you can consider, for instance, is how the time evolution of this probability of having A, where this first term is, where this S is like the selection coefficient, and these is like the probability. These are the probabilities to go between the alleles A and B. Once again, but these are just like rates. They're like doesn't tell you how the mutation happens. And I guess to some extent, it's maybe implicitly saying maybe how the mutations happen doesn't matter. And so in this talk, I guess I want to convince you that, well, how mutations occur can change the eco-evolutionary dynamics. And that's sort of the motivation for this talk. So not only how often do they happen or the tempo, but the mode of mutation can cause different eco-evolutionary outcomes. Okay, so in this talk, I want to consider two different types of mutations among many, but these are just two. This is sort of a proof of concept. This is not a fully sort of extensive framework. So I'll consider two types of mutations. One is external. So you imagine there's some microbe that has one chromosome, and because of radiation or some external factor, they get a mutation denoted by this X. So that's one mode. Is this it's external? Another one that I'll consider is replicative. So imagine during the cell cycle, you make a replication error, and then when you replicate your chromosome, one of them will get the good chromosome, the other one will have the one with the mutational defects. And this is another model. So I'll consider these two main types, and what I'll show you is there's different eco-evolutionary outcomes between these two. Okay, so let me just first motivate on a population level, a thought experiment, what you would expect if you had these two cases. So if it was just external mutations, then you would expect that, imagine you have two ecotypes. Ecotype A is very large, but relatively constant. And then ecotype B is small, but is growing very fast. So if you just had external mutations, you would expect that most of the mutations would happen on ecotype A. And if you had case two, where most of the mutations were replicative, then you would expect this to be most of them to happen on the fast-growing population. This is just sort of a thought experiment. How they, in reality, you probably have a mixture of both, and others, but it's just sort of an idea. Okay, so let's just keep working on this. So what does this look like mathematically? So let's just first introduce this landscape. So this is I'm just showing you in 1D as like illustration, but this framework that we've worked out is true for any dimension. It's the model is for any dimension, but this is just the idea. So imagine that there's some phenotype coordinate, and the idea is the phenotype coordinate could be, if we look at the bacteria and phages example, for instance, this is the bacteria would be the resources, and the phages would be the consumers. And the idea is like, well, if there's some phenotype coordinate, that could be like the proteins expressed on the bacterial wall, for instance, that would allow a phage to eat it or and infect it. So this is kind of the idea. It's like it's the different phenotypes or genotypes that allow. So if they're on the same one, then this would be where the phage would want to infect the bacteria. That makes sense. So here I'm saying there's some lattice, which we'll eventually take to the continuum limit, so it'll be a field. But the idea is that the consumers will are here, and I'm just showing it going down, but really they're both positive. But this is an arrow, so both would be up here. But the idea is that where you have high consumers, you'll have less resources and so forth. So the dynamics of this would be there's birth and death of consumers. There's mutation. So the only way that you can change on the phenotype lattice is in this model is through mutations, whether it be exogenous or replicative. And then the resources can also grow or be consumed. And so that's sort of this is the one. Yeah, this is the model. And so now let's look at what this would look like from an equation. Just first the mutations. So external mutations, basically can be represented as a constant rate. So it's just some constant rate that's proportional to the population that you'll have an external mutation. And you could just write this as like a diffusion term with a rate mu. I will just say there's a caveat here. So in the paper we talk about this caveat, but I feel like it was not like good enough to spend like 5 minutes talking about this. But basically this would actually be like they would have higher order moments. So this could be like ballistic motion and things like that. But there's some quantitative regime when you can have this. But because there's like kernels to jump long distance, it's not approximate. It's not always diffusive. But let's just, those are sort of, if you want to know the details, I'm happy to talk about that. But I'm just sort of giving you a qualitative idea. So imagine this is just being like a constant rate of diffusion. And then, but for replicative, this is saying that there's some rate 1 minus p that you will get the good chromosome, and there's some rate p that you'll get the bad chromosome. So this is just some probability between 0 and 1. And so this is like a nonlinear diffusion. So imagine f is just some nonlinear functional of the field chi. And so you could probably expect that there would be differences here. But so these are the two I'll consider. So now let me just show you the full model. First, I'll just show you I kind of reiterate what all the interactions are. So for the resources, they have this self-renewal, and there's some growth rate. And there's also self-limitation through some carrying capacity, kappa. And for the consumers, they can die, and they can have consumption. So they'll eat the resources, but there's two main things. The first one is where they consume the resource and they grow. So it's the energy from the from eating goes towards their growth. Another one is where the energy, when they eat that, it doesn't go towards growth, so it goes towards everything else, like metabolism, their immune system, and so on. And so this is how, by the way, all of this kind of, so Z1 is Z1 and Z2 are the prefactors, and psi and phi are like some memory kernel that's we'll take to be exponential, but it doesn't. The analysis is done for like a generic kernel. But the idea is like it's just finitely peaked, and it's just you're most likely to eat the resources that are right next to you, but there's some finite decay. So we don't consider anything like crazy, like like the hat potentials or things like that. Okay. So in equation form, this is what you would approximately get. And so the first terms here, so these are the mutation terms. This is the birth from consumption. This is the wasteful consumption. And for now, I'll introduce noise in the later part of the talk, and this will be demographic noise. But right now, I just want to first ask the question: what is can we see some phase transitions in this model in any dimension in the mean field model, so without the noise? So it turns out that there is phase transitions only so exogenous does not, so the external mutations in the mean field does not have phase transition. So that means we couldn't find any instability for positive definite memory kernels. It's only for this nonlinear diffusion or this replicative case. Okay, so this is what I'm showing you here. So for the replicative case, if you'll have so here Q is the wave vector. So you have some finitely peaked. You'll have some finite patterning as proportional to square root of 2 divided by p, where p is the probability of having this replicative error. And here's a phase diagram actually. And so as a function of beta is just p over 2d. So d is the dimensionality, and so this is a phase diagram for all dimensions, and p is that probability, and over here is what we're calling the scaled relaxation rate. But it's just when you non-dimensionalize the equations, it's a formula that comes out, a relationship that comes out in terms of gamma, the death rate, and Z1 and kappa. And here you can see that for a lot of the phase diagram, you're homogeneous, in which we quantify through if you plot the determinant, then you'll see that there's nothing that really diverges. If you see a divergence in the determinant of J, that'll tell you that you have this Turing-like patterning. But actually over here, if you look at the determinant of J, so basically we linearize those equations that I showed you, and then then you get a Jacobian, and then this. So the idea is when you see a divergence in the Jacobian, that's telling you that you get this patterning. Now here's this is this caveat I was telling you about. So you see this red line. This is related to the lattice spacing, or this that kind of is in here. And so if this divergence would have happened down here, then you would have had to consider this like transport, or this like nonlinear, or it wouldn't be diffuse. You couldn't kind of approximate this as being diffuse. So it has to the patterning kind of has to happen between some like larger length scale. But anyway, that's besides the point. But the idea is you can get patterning, and actually we simulated this, and so this is showing you in the lattice version. So it's a simulation with the fluctuations, where you get this is the homogeneous, and then in the patterning phase you get something that's like a Turing instability, where you get, this is just a 1D, and so you get these peaks so the resources are this, and the consumers are here, and so you get these peaks that form in this instability. And I just want to reiterate here that this is solely due to the eco-evolutionary interactions. So it's not like some phenotypes I don't make any of them special or better or more. There's not really an aspect of fitness here. It's just saying that this patterning or this inhomogeneity is happening just because of the interactions between ecology and evolution. Okay. But this problem, this has a little bit of an issue, because this only happens in one dimension. So we tried higher dimensions, and as you can see here in this phase diagram, to get to this high, if you have like 2 dimensions, for instance, even if p can only be 1, so even in 2 dimensions, you won't ever be able to reach this small area where this patterning happens. And so that made us think, like, how can we, in real systems, nothing is going to be one-dimensional. Most it's thought that a lot of these are higher dimensional uh, processes. And so that made us think. And so one thing that we found is that if you add a term related to this, what we call host defense, this is basically saying, well, bacteria have an immune system, and there's some probability that if the phage infects them, they'll be able to fight it off. And so now let's consider a more general model. And so this is saying you have all the same interactions as before, but there's one more, which is this toxic consumption. So you have consumption, they grow, so the phage grows, for instance. So the phage is now is still a consumer. So the phage can like inject its DNA and then grow, or it can inject its DNA and nothing happens, or it can inject its DNA and it fights it off. The bacteria fights it off. And so now in this model, what we found is that when you do the same type of analysis, now you can get patterning in arbitrary dimensions. And the phase separation is much more expanded. So here's just showing you another phase diagram, and this is just showing you how the instability grows. And if you look in 2D, you can get these patterning now. So this is now just a 2D coordinate. I'm not quite sure why it's not as uniform. But anyway, this is where just showing the patterning and not patterning. And in the last few minutes, I just wanted to also mention now noise. So that was all deterministic, but now the question is, what is the effect of noise? And in this model, the noise would be demographic noise. So there's 2 instances where noise can matter that I can think of. One would be, of course, large dimensional space, but finite populations. Another one was, of course, finite dimensional systems. So the idea is that there's some upper critical point, which we haven't calculated yet, where it would be a mean field model and the noise doesn't matter. But if you're below that critical point, which, maybe, I don't know what the critical point, but, for instance, like Landau-Ginzburg, it's 4. So if you're below that, then noise would matter. And there's some indication that there are some, like maybe like the flu virus, there's some that believe that even though it may be high dimensional, there's some effectively low dimensional space that could potentially be like 2 or 3. So this is an interesting problem for that. So the idea here is, what is the effect of noise in these models? And so for this, we took now Q is the wave vector. So this is saying we took that model before, we write a path integral for it, and we've write it out to quadratic order. So it's going to be a Gaussian. So the J is the Jacobian and B is the noise contribution. And here the conjugate variable is, like this is like the MSR-JD formalism for path integrals, where you have the conjugate variable x tilde. Here x is just a vector for chi and rho. And so here we can actually write the power spectrum. So this is the fluctuations of the consumers. And this we can write like this. And so basically you can compute this integral because it's a Gaussian integral. And then you can look for finitely peaked. So within this power spectrum, you can look for finitely peaked peaks. And that would tell you about noise-driven patterning. And so for this is still some preliminary, but the idea is that now if you look at that same phase diagram for beta and eta, so this is the same one as before for the 2D. The idea is that this is the deterministic line that I showed you before. It's not painted like it was before, but here what we're finding for that same replicative, or with the toxic defense, that you get actually noise-driven patterning that lowers this line almost to 0, where it's like it almost makes patterning like generic in this model. But also what we're looking at is there's other classes of models like the exogenous, where potentially where we didn't see the patterning, noise could cause that pattern to form. So we're still working out the details of this. But yeah, the first step would be sort of quantify this. And then, so I haven't gotten to the very extreme, but we're also looking at the large deviations, which are, of course, very important to this problem. So with that, I just want to kind of briefly mention how this could relate to real systems. Obviously, we're still, the phenotype space will never be able to be quantified exactly, because, you just won't be able to quantify, like, what is a lattice site, so there's too many things that you can't quantify. But the idea is, can this help us understand real systems? So for this, for one system, prochlorococcus, they have hundreds of discrete, strongly diverged genomic backbones that can coexist within a single mixing volume. So this could potentially help understand like how that patterning can exist. It could also maybe help us understand cancers, where exogenous processes like smoking or replicative processes like homologous recombination-based repair errors can leave characteristic signatures in the spectrum. So these are areas where, maybe once we get a better handle of this, we can know how to apply it to data. But yeah, this is the main problem is this phenotype lattice. Like, how do we address this without, like, how can you make predictions without knowing what that lattice is, basically? So in summary, I just wanted to summarize that. So I showed you this consumer resource. Well, I didn't say consumer resource, but it's an eco-evolutionary model that shows that you can have patterning and how mutations matter. That some can cause this patterning just from eco-evolutionary interactions, and some don't. Yeah, and so I also showed you that though, if we don't consider host defense, that in the model we didn't see pattern. You can't get patterning in higher dimensions, but if you include this host defense term, you can get patterning in arbitrarily high dimensions. And then I showed you how noise can expand the region of patterning, and also just suggested that it can also work for like the external mutations where you didn't get patterning in the deterministic dynamics, but the noise could potentially cause it to happen. Okay, so with that, thank you for your attention, and happy to take questions.
So, Trevor, could you help me get some more biological intuition? So when you say patterning in the trait space, I'm imagining something like you have predator fish, prey fish, and this fish gets to be 1 meter, 3 meter, 5 meter, 7 meter, whereas the little fish, the prey fish gets to be 2 meter, 4 meter, 6 meter. Like that's what I saw. I think it was like complementary patterns, right?
I see. Yeah.
So can you help me? That's my first question. Can you help me get some biological intuition about this? My second question is, how does type of mutation matters, you said. How does that actually play into this patterning? I couldn't quite follow that also.
Yeah. So okay, I wouldn't necessarily say it's like a direct one. Like, I'm not going to say I can understand, guppy fish or bacteria phase, but this is more of just, I would say right now it's more motivated. But the idea is, if you start, I guess, or maybe a thought experiment would be, if you start out with genetically identical, or with slight variation of bacteria and phages, and you get patterning in an experiment, and you see, then after you run it for some time and you sample you sample that population, you're going to get variations, right? And the idea is, like, where does that come from? And I guess this is just showing, well, you don't necessarily have, I'm sure there are actually some genes that are definitely they're gonna be like predisposed to have. But there's going to be some that maybe it's just due to eco-evolutionary feedback. So this is saying that you don't need to have sort of like favoritism in certain genes to get sort of like genetic variability. You can actually get it just from eco-evolutionary feedback.
But the complementarity of the patterns, like
Yeah.
one is high, the other one is low. You don't have a good intuition for this, maybe, do you?
Not necessarily, no.
Okay.
Yeah. Yeah, I forgot your other point, but
Can you give like an example of what the genotype space might look like?
Can you say it one more time?
Sorry, can you give an example of what this genotype space might look like? Like, do you have any sense of like what sort of like phenotypic space?
Yeah, so I was trying to illustrate this with the bacteria and phages. We're saying like, there could be some expression of proteins on the surface, for instance, and then one mutation you know, to those proteins could drastically change how those proteins are expressed, and that could potentially cause that phage to not be able to infect it anymore if it jumps somewhere. And vice versa. So the phage, if it changes, you know, how it attaches to the bacteria, it that could change whether how it if it can affect or not. There's different proteins, for instance, like lambda phage. There's different proteins that from how it latches onto it, how it recognizes the host. So there's a lot of different ways that you can have. Yeah.
Have you like thought about, sorry, have you thought about like sort of the evolution of mutation rate itself? For example, like mutator phenotypes?
No, no. Yeah. But this will be interesting.
Can you give some intuition for the noise-driven patterning? Like if you did some sort of small noise approximation, is this like you have sort of all stable wave numbers, but then maybe the least stable are the ones that get excited by noise? Is that the kind of thing that's going on in these?
I think so, yeah.
But-
I mean, so yeah, so once again, like most times when you consider ecology or eco-evolutionary systems, it's usually assumed high dimensional, and it like to the point where noise doesn't matter. Once you're above some upper critical point, the noise doesn't matter, like unless the population is really small. And so I think most kind of sweep that under the rug and say it's probably high dimensional. It doesn't matter. We're assuming large populations. But yeah, so when you're below that critical point, the noise matters, and you can't like sweep it on the rug. It's there. So the idea is, yeah, but yeah, this is
And
it.
have you looked at your J plots as like the one that goes down the farthest? Is that typically the wave number that gets excited?
Oh,
Or can you compute those J plots still?
so actually, so the power spectrum has it might be different because the power spectrum is a fraction, and the denominator is basically the determinant of J squared, but the numerator has noise terms. So basically, you'll get noise-driven patterning if your noise term also depends on the wave vector. So if it has like q squared or higher, then you can get patterning. So it depends on the combination of both. And when you take the derivative, yeah, so it could be proportional to it.
Oh, but it
Yeah.
could shift it to some
Yeah.
different pattern or wave number.
Yeah.
Cool.
Other questions? No, let's thank Trevor again.